18 months of analytical data, a clean peptide map, mass specs down to the individual glycan, and deficiency later still come back asking one thing: how do you know the protein is folded the same way? This is the question circular dichroism exists to answer, and most teams learn it was expected only after somebody asked. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to build higher order structure and comparability packages that survive review. Today, when does FDA actually expect circular dichroism data and when does it not? Before moving forward to our discussion, if you have found anything valuable in this channel, please like and subscribe. This gesture of appreciation means really a lot to us. Let's move on to our technical discussion. So, protein drugs are the only medicines where the molecule shape is part of its identity. Get the sequence right and the fold wrong, and you have a different product. One that can lose potency or provoke an immune response. Regulators worked that out during the first wave of well-characterized biologics, and it went into writing in ICH Q6B, which named higher order structures something you characterize rather than assume. Q5E followed for manufacturing changes. Then the biosimilar pathway opened, and proving that two proteins fold alike stopped being academic and became commercial. So, what is the instrument doing? Circular dichroism passes left and right circularly polarized light through your sample and measures the difference in absorption. Chiral environments absorb the two unequally, and a folded protein is a stake of them. Below 250 nanometers, the peptide bond dominates, and you read secondary structure. Alpha helix gives twin minima at 208 and 222. Beta sits a single tough near 218. Above 250, aromatic side chains and disulfides take over, and you get a tertiary fingerprint. 20 minutes, a few hundred micrograms, no cluster, no labels. Far UV and near UV circular dichroism are two different experiments, and reviewers read them differently. Far UV, roughly 190 to 250 nanometers, reports secondary structure content, how much helix, how much seed. Robust, reproducible, and blunt, a protein can lose a critical local fold, and the far UV spectrum barely moves. Near UV 250 to 320 is the sensitive one. It reports the packing around tryptophan, tyrosine, and disulfides. So it catches tertiary changes, far UV misses. Submit only far UV, call it a higher order structure comparability, and you should expect a question back. The pair is the answer, neither only one. So there are three places this test reliably shows up. 1. Characterization of a new biologic. ICH Q6B puts higher order structure in the physicochemical characterization section of the dossier, and circular dichroism is named there alongside NMR and crystallography. 2. Comparability. Change your cell line, scale, site, or formulation, and Q5E expects evidence the molecule didn't change with it. 3. Biosimilar analytical similarity, where you compare against a reference product across multiple lots and bar is highest. If that breakdown clarified where your own program sits, a quick tells me keep making these regulatory maps. Whether FDA expects circular dichroism from you depends almost entirely on what you are developing. So here is the honest tiring. Filing a new therapeutic protein or a biosimilar, treat it as expected. You will be asked for higher order structure data, and CD is the cheapest orthogonal way to supply it. Post-approval change or chiral small molecule where absolute configuration must be established. Situational, electronic and vibrational CD do that job. And ICH Q6A's chirality decision trees tells you when it matters. Routine release testing, almost never. Circular dichroism is a characterization method, not a specification test, and that distinction saves program a lot of wasted validation efforts. Now, circular dichroism packages that get questioned in a review usually fail for three reasons, and none of them are about the physics. First, the single lot comparison, one reference lot against one test lot, offered as proof of similarity with no sense of normal lot to lot variability. Second, an unqualified method, no signal to noise, no calibration check, no accurate concentration measurement underneath it, and CD signal scales directly with the concentration you assumed. Third, overclaiming, structural equivalence declared from spectra that overlay by eye with no criterion defined. At Resolve Mass, we have rebuilt higher order structure packages that were scientifically sound and still got queried purely because nobody wrote down what comparable means before the data existed. Which is why a defensible package starts before the instrument is switched on. Define the question, identity, comparability, or similarity, because that decides everything downstream. Qualify the method for that purpose, linearity, precision, signal to noise, and the sound concentration method beneath it. Run reference and test material in the same session, same buffer, same operator across enough lots to show real variability. Set your comparison criteria in advance. Then report alongside orthogonal data, DSC, FTIR, or intrinsic fluorescence. CD alone is a claim. CD plus one orthogonal method is evidence. So what should you walk away with about circular dichroism in today's session? It reads folding, not sequence. Far UV for secondary structure, near UV for tertiary. Regulators expect it in characterization, comparability, and biosimilar similarity work, and almost never on a release specification. Its weakness is resolution, so it travels with orthogonal methods and define what comparable means before you run the sample, not after. If you are building an IND, a biosimilar analytical similarity package, or a comparability protocol for a change you have already made, and you want a CRO or CDMO partner that for this kind of projects, Resolve Mass Laboratory supports small and mid-sized pharma teams from method qualification through submission. Thank you so much for watching this video till the end. Before you go, I want you to do three quick things. Number one, subscribe this channel right now, because the more you subscribe, the more valuable pharma insights, expert tips, and industry updates we can bring to you. Second, please comment and let us know what did we do wrong and what did we do right, because the more you give us the feedback, the better we will be able to make videos for you. And number 3, share this video with at least one person who would find it useful. You never know who is stuck on exactly this kind of problems right now. I will see you in the next time. Until then, goodbye for now.